bongo. All right. Ready to go.
3, 2, 1.
Locked.
What's the sample number on that?
101.
101. And that was
Skin number 1.
that's Skin 1.
Roger. Flying the vehicle with buttons.
So that's a water sample that will be filtered back on the ship. Have you been doing that, Taylor Ann? Has that been your kind of one of your many jobs?
Yeah, so I try to switch off with our other data logger, Mary. So we switch off like every day of sampling, from filtering to doing biology, helping science manager Steve with the biology. But yeah, I get a lot of filtering done in that lab for my microplastics and DNA and particulate organic matter as well.
Oh, so all of those go through the filter as well, not just the eDNA.
Yeah, so we have different setups for each one of them. Usually we do the eDNA first, since, you know, we got some DNA in there that we want to get filtered right away. We keep it in the fridge and then filter them, and then we get to the other water to filter. Doesn't take too long. Although I recently changed my filter sizes to 0.4 microns, and that takes way longer to filter liters of water through.
Yeah.
So, yeah. But that's all right. Still, I can see like microplastics, mostly fibers, with my naked eye. So that's pretty interesting.
You kind of get a preview as to how much there might be at a local site. But something you don't quite realize till you start doing it is the importance of just keeping track of all the samples and being kind of consistent and having a good notes taken in the field, because you just rely on them so much later. And as I learned, helping Meredith out, not dropping the water samples on the way from the fridge to the filter.
Oh man, that hasn't happened to me yet, knocking on wood.
Yeah.
Sure hope not.
Felt quite bad.
Yeah, I've spilled my own samples, but that's okay. I'd rather spill my stuff than anyone else's. So.
Somebody else's.
Yeah, but that was the first day when I was, you know, getting into the groove of my routine.
Yeah. Yeah. And the other thing it's easy to forget as a scientist ashore is how much the boat moves.
Oh my goodness, yes.
Yep.
The other day I broke my glass graduated cylinder when the boat just decided to tip me forward. Thankfully I didn't cut myself on it. But
I see you with those big the
big glass jugs.
Jugs walking around.
Juice dispensers.
I saw you with 2 coming back, and I was like, ooh, that's
risky, huh?
Risky, playing a dangerous game. But.
Yeah. I'm having a look on the science chat, and we're getting some feedback from Lizzie, just saying that we're sort of on a quest for hard substrate. So whichever speed the pilots feel you'll feel comfortable with, we can keep cruising north, and then probably we'll do another transect when we get a reasonable distance away again.
Sure.
Thank you. Yeah, I'm pretty sure it was this crew, wasn't it, when we went warp speed southwards.
Oh yeah, over the for the Petrelli Soul. Yeah.
Yep. It was worth it. I'm very glad we got back down to that site.
Yeah.
Got those images from the last transect.
Got something coming up in sonar.
Oh,
Solitary target. Yeah. Straight ahead.
another little sea pen. Small grenadier. Little rat tail, that fish that swam past.
It's gonna be trash or a rock.
Here it is.
There's our target.
You
Target.
say rock. Nice. It's a little habitat island. Yeah. We got some dead glass sponge, some dead barrier by the look of it. Hagfish, thorny head. I guess there's a crab underneath. I don't think there's anything to sample on this one. Oh, there's one of those little sea pens though.
Nice shot.
Yeah, nice shot of the hagfish.
Probably turn before you get tugged. That way you'll have your full, you won't be like fighting it. You know, you can just zoom ahead, go to like 45.
Could we get high back up on channel 3, please, Stephen?
Sure thing.
Thank you.
Bridge nav.
The spot.
Add one step, 100 meters north.
Oh, look at that. Have we got time for a little zoom on this one? I think this is one of those, it's one of the fiber optic sponges. It looks like it's dead, but has got either another type of sponge growing on the stalk, which is something we know happens. There was a study where they looked at the colonization of dead Hyalonema sponge stalks, and then they also put out some artificial sponge stalks and compared kind of colonization rates on those two different substrates, and they found that different things would attach to the artificial stalks, to the kind of sponge stems. Because the stalk is made of glass, it sticks around for quite a lot longer after the kind of the living bit of the spongy tissue at the top has died. I can't tell on this one actually if this is a different sponge that's growing on the stem, or it actually looks like it might be one of the fiber optic sponges that's just on its way out. Let's have a look. Oh, maybe not. It's good to see though. We might come across some healthier ones shortly. Thank you for the zoom. That's fab. Another little sea pen in the right there. Maybe one of those little snakehead eel pouts. Oh, and another one. Oh, it's something big just took off there.
Dragging that right. Yeah, do you want to stop for the kelp?
I always feel bad asking, but yes.
Taylor, if you now is a good time. If you
Yeah.
want to stop for the kelp,
Let's do it.
we're not on transact.
Hold position. Thank you.
For anyone joining, we're exploring about 1,000 meters. We're south of Santa Cruz Island, off the coast of California in the Channel Islands, and we've just stopped to take a kelp sample.
Yeah. Yeah, you can look at, just glance at your DVL track too to see where you're at. So. Is that it? Is that a cucumber? I'll back him up. Bridge now. Two 0 meters south. Was that it, or is that a holothurian? I don't think so.
Holothurian.
Might just be north in the dust cloud.
we go. It's here somewhere. Go very far.
Sure, you just want to look up the other way. Zoom in here. Right on it. Right on the trail, just about.
Thorny heads and sea pens.
Might as well go as far as your leash can take you, then turn around.
Sure, we'll see more.
It's lovely.
You could also face the slope. Yeah. No, just yeah, I didn't. I should have dropped the target, but I didn't know we'd fall off it. Just in sight. Yeah.
Well, we'll look out for the next one if we don't
Yeah.
come across it as we come
Yeah.
back up here.
We'll see more.
Maybe if you back off the slope of it, you'll get some more view. I think you're kind of dragging on it. There's your mark from when you came last time. So that's how you know you're. Yeah.
All right. Did we go right over it, or was it off to the side?
I thought it was slightly off to the left.
Yeah. They come up and north, or sorry, upslope a bit here, but yeah, and you only have a very small field of view because you're up against the slope. Yeah. Give us some perspective.
Oh, is that a bamboo?
I reckon that might be another one of those dead sponges, just the stalk.
Sorry, I backed the ship up. I can go back north now.
All right. Okay. So Renato, I've just had some feedback on the science chat as well. So on looking at HIPAC, between where we are now and BG1.
I'm sorry, hold on a second there, Abby.
Oh, yeah.
Okay, say again.
So just looking on HIPAC now, between our current position and BG1, slightly to the north and about halfway between us, there's a yellower zone on the map.
slope. Yeah. Yep.
Yeah. So we want to make sure that we go through that point, and probably taking either going north and then cutting west, or a more diagonal, whichever way you think is going to give us kind of more slope, more chance of hard substrate.
Okay. Maybe we'll just go straight there along the contour.
Yeah, that was good.
Yeah. Not yet. Yep. Bridge nav. Step north 20 meters. Okay, so I'm gonna go after we get this bump north towards you. I'm gonna go 305. I'm moving north. It's moving. It's moving north. Called it in.
And I'm back.
Oh, hi Meredith.
What's that?
Sorry about that little interlude there with working with had a meeting with the applied physics lab talking about our fancy sampler we're working on.
I see what
Is
happened,
this the
Dan.
one that you're thinking might?
Pardon?
Is this the oh, I just lost my video again. Is this the sampler that would be autonomous?
Yes. Yeah, this is the autonomous eDNA sampler.
Very cool. Renato is currently, he's setting a new course for us to make it to that big yellowy patch on high pack. You think might be hard
Excellent.
substrate, kind of midway between where we are now and point BT1.
Awesome.
And we just failed to pick up a piece of kelp.
Yeah, sorry about that. I lost it in my own dust cloud.
Yeah,
No.
right.
There'll be more.
I saw just out of the corner of my eye, you found a big Hyalinema sponge.
Yes, yeah. I think now it was well, either that sponge was dying or that was a different sponge growing on the stalk of a dead Hyalinema populiferum, the fiber optic sponge.
When we were up in Olympic Coast, we found some that were definitely Hyalinema populiferum, but their stalks were tiny. And so they looked like they were some other sponge, like possibly the Acarnis. And then we went to sample it and it was just glued into the
Right here.
substrate. We finally worked it up enough that you could just see the stalk.
Ah. See, that's really interesting. I remember asking Henry if one of those fiber optic sponges was the Acarnis, and he just laughed and said they were about as different as you could possibly get. So I felt kind of embarrassed by that, but yeah, I've never seen them with the like short stubby stalks like that before. I mean, they're both orange and they got kind of flat tops and smaller individuals.
Yeah.
Just bit of a different depth zone.
Abigail, I just had a thought about your bandwidth. If I think you're talking on the Nautilus feed as well as the SPL, and if you want to just go to SPL, I think
Yes,
that might
I
help you
am.
out a bit.
Okay, doing that now.
Cheers, thanks.
Thank you.
All right, now there's a piece of kelp with some urchins on it.
Wow, that's like a jackpot. But if we could snag that, that's
You okay with that, scientist?
Yeah, that would be great.
Bridge
Yep.
nav?
I come down ever so gently.
This one has urchins to pin it down.
Hold position.
Oh, they're so
Thank you.
tiny. I don't want to take their food, but I also would like to collect the urchins too. They're feeding on that kelp.
Maybe if we get lucky with the grab, the urchins will come up on the
So like,
kelp.
just turn my heading a bit. Rudder. Yeah. Try not to thrust too much to get all the sediment up. Yeah, it's not really going.
And Taylor Ann, would you like to get sediment
there?
cores here as well?
We could.
Up to you.
Yeah, I would
Grabbing
love
the
that.
kelp. Could get the trifecta.
Seems like the left
kelp,
corner
the urchins,
kind
the
of in.
sediment.
Yeah.
Seems like the left
It's a
corner is kind
jackpot.
of in there, but
I discovered what some of the source of my V-Link issues were once I was back to doing this kind of midweek, and my lovely spouse was not home sharing my bandwidth.
That'll do it.
Ha.
I'm gonna disturb it all.
And if we could get a zoom once the little dust cloud kind of dissipates, that'd be cool.
Bridge nav. $10 meters, 030.
Oh, grab it.
These are tiny urchins.
Yeah, they're really tiny.
Is the plan to slurp the kelp and the urchins, or just the urchins?
I'd like all of it. Well, not all of the
I
urchins,
just
but
meant the method, sorry.
oh, yeah, I don't know what would be best. I ideally would like to slurp it. That way it'd be isolated, and not mixing and sloughing off water. So like that way I can also filter the water from the slurp.
Seems like I'm drifting away a bit.
Yeah.
But I know it's kind of touchy to slurp kelp and maybe those urchins too. So
I gotta get up a bit.
You need me to come wide?
Yeah, can you come wide for a second? I'm sliding down the slope. Yep.
Creeping slowly backwards.
Oh. Yep. Too close.
Super fine grained mud here.
yeah.
Yeah, I'm keep sinking in and then sliding. All right.
You just had to make sure to get the, make the sampling as challenging as possible.
Great. Yep.
So does that look like a frond from the top of the macrocystis?
Yeah, it does. Those characteristic wrinkles in there.
So for those listening at home, macrocystis is the giant kelp that forms the kelp forests in places.
Yeah, ready to flush. 6. T6. Oh. No, don't move, kelp.
Uh-oh.
Oh no.
Yeah, put a little more on my verts, maybe. I could put a little more. That's on my.
So all the jars are empty except for slurp number 1. So any of them besides 1. Yeah.
I think someone is off SPL.
Oh,
Probably Dan.
it's probably
I think
Dan.
it's Dan.
It's like, I have a feeling Dan is talking, but
Yeah, you don't want to listen to me
Only-
curse, so I'm off SPL.
oh,
Okay, you can zoom
good
in a little
reason,
there
good
for him.
reason.
And I'm going to start it up there. Jar number 1. Zoom in a bit for him, Steve. That's good. Okay, got a hold of it. I'm at 40%.
40%.
Yeah,
Little nook is stuck on the outside of it, I think.
I give you a little more jam here. 50%. 60.
There it goes.
It's going in at 70. 80%. Okay, zoom out a bit, Steve. So if you play with the hose a little there, it'll help it get around the bend.
Oh, wow. Look at
Yeah.
that.
Oh,
Yeah.
nice.
Hey, I'm up back to 0.
Nice.
You want some of the little urchins too, I guess?
Yeah. It'd be great and just slurp them up.
Okay, if we get close to your target, then Steve can zoom in. So. Ready?
Ready.
Suction is on at 50%.
See the mud shaking.
Or the little opioid.
Yeah, a little brittle star went up.
Yeah.
Just gonna pan your camera a little to the left, all right?
Rudder.
That was a little too
Yeah,
much.
that's good.
good.
I don't need all of them.
They're going up pretty easy.
Might be in the mud.
See him coming in there?
Not yet. Oh,
There's
there's
one.
one. Oh, there's a cloud of mud, so I'm sure they're in there. Let's see.
Gonna turn up the jam for their little spikers that catch it. I'm gonna turn it off for a second and see what falls down. 1, 2.
So while watching last night, I saw a tape mark on the hose. Is there some magic to that tape mark?
Yeah.
That's the danger mark. And then
How
there's
many did you suck up?
there's a red mark after it, which is like,
I think you got 3 or 4.
yeah.
3 or
Right,
4.
suction back on.
Yeah.
You could probably put the hose away now. Or get ready to put it away.
So is there a danger that you extend the arm so much that it
Hose comes out.
disconnects the hose?
Yeah,
Yeah.
more so the hose. Yeah, the hose will.
There's a lazy S. The hose is attached to a bungee, and there's a lazy S in the underneath the vehicle. If you pull it too hard, it will either kink the hose or pull the bungee out. Or I can't
No more
tell how many
flaps.
we have there. Just gonna turn it off, and then I'm gonna bump the jar.
What am I doing wrong here?
There's at least 3 in there.
At least 3.
Yeah, that's good.
They're already jumping onto the seat to the kilt. So. Okay. Now the hard part, parking that hose,
Yeah, I'm all out of sorts right now.
snaggletooth jaws. Yeah. It's a 6 function now.
Yeah.
And which slurp jar number was that? I can't quite see from the...
The jar number?
Yeah.
It was in jar number 2.
Okay.
Need to make that a 7 function arm again.
Yeah. Oh, there we go.
I can just grab the nozzle if that's easier instead of the handle.
Outside of the biology world, would these cutting jaws just be used for like cable cutting and that kind of thing?
What's that?
I said outside of the biology world where we call these jaws the coral cutters, are these just a cutting pair of jaws that would get used for cables or other materials?
Nope, they're just used to make the ROV operator's life absolutely miserable.
Yeah, I can see why you dislike them.
See why I do not favor them.
Yeah.
Did I hear you wanted push cores?
Yeah.
Oh yeah, we're in push core land. I have to push buttons then. What do I need to do? Cameras.
So does whoever is piloting Herc also operate the arm, or can you sort of decide amongst yourselves, could the Argus pilot also, can you take it in turns?
Yes, I'm at least a few factors slower than Dan. So.
And give it a few decades. I'm gonna do that button thing here again.
Spin around, you're saying?
Why don't you come out of there and find a undisturbed location. Center your camera up, but still look at the front of the vehicle. Could probably slide to your left up the hill some. You're definitely going to want to be more southeast on the heading, because you got the long jaws on there, then the core tubes are going to be long.
Southeast. All right.
Yeah, so you want to bring your head to the right quite a bit. You know what I mean?
Yep.
You're basically going to have a baseball bat in the end of the manipulator, and you're on the hill, so you won't have room between the bumper and the seabed to get the sample. I'd come up a little more. We dusted it fairly there. Maybe up and to your left. So you're, because the current's in your face now, so
Yeah.
forward on the
Now I'm
ROV
dragging.
should be, in theory, undisturbed. Could see an Argus.
Ooh, little profiler.
Yeah, okay, nice and easy somewhere there. Oops, what did I do?
Wasn't very easy, but it'll do.
There's, as far as we know, nothing in the starboard sample box, correct?
No, there's nothing there.
Okay.
Mudstorm.
What's that?
Mudstorm.
Yeah, it looks worse than it is. Look.
Until there's less current on this dive than on previous dives, the way the mud is just hanging around.
It is pretty dusty there. Let's try one more time to come forward a little. Gently, gently. Just leave the arm right where it is. Turn your Z bias down a little to like 30. Shell float up nicely for you.
Oh,
Other way.
I'm on the wrong,
That not be there.
this guy, though.
Huh.
How do I get back to the...
That's all right. Watch it there.
All right.
Turn it to, yeah, see which way you're going to turn it. So turn it till it's like 30-ish. To the right, I think. I turn it down a little more so you don't have to thrust up. That'll let the vehicle float up better. I'm going to look back down for you, or you can look down at the front of the vehicle. Yeah. So you can see an Argus if you go like a vehicle length, it's clear water. That should hold you. Beautiful. Might need just a little more down because when you push the core in, it'll want to lift the vehicle up.
A little more down. So.
Yeah, I would go for like 50% maybe. Just ease it in. If you go too much, it'll start rolling the vehicle. But yeah, that's better. It's embarrassing to have it float up when you're stabbing the core.
Anyone watching at home, you can see a nice view in channel 3. You can see the claw about to, Jake about to grab hold of one of the push cores mounted on the starboard side of the vehicle.
You could zoom in on Argus if you wanted to, Steve.
Can you do the sample salvo so I can see it just a
Oh yeah,
little
sorry,
better?
sorry. Sample salvo.
And we already took push core number 1, so we can go for number 2.
Number 2, roger.
Nice. I'm already right around that one.
Oh, zoom a little more there. Centered it up for you.
Ah. Oh, the wrong one.
It might be easier for you to get the back one.
Number 1?
The one furthest away.
That's what I'm doing wrong.
For those watching along at home, if you look at camera 3, you can see the push cores being grabbed from the side of the vehicle.
So as you swing around, swing easy, because
Yeah,
it would be easy to swing into the hill there. I'll go pan right a little more for you so you can see the minute.
Is they're good?
Yeah, but move to a new spot because you dusted it when you twitched the...
There?
Sure. So tailor in.
Yeah, that's great.
Zoom in just a little bit for him, Steve. There we go, just a touch more. It tends to, yeah, it's probably good.
All right.
The core tends to slide out just a little before the valve closes. Okay, Steve, you can zoom out now.
Now for the hard part.
Just take your time.
Nice.
You want to try and keep it horizontal if you can as well. Steve, zoom in. a baby.
Oh, it's a tiny sea pen, or one that's half retracted. We're pretty sure this is Stachytilum, although that's a member of a group that you really have to have in hand to really tell, because it's got a very similar genus called Protoptilum.
Somebody's breathing into the mic.
That's me.
Is it?
Yep.
I was going to say, actually, the only acceptable person it is to do that is Jake.
I knew it was Jake. I was going to wait till he was back.
My
He's-
mask fell down and my nose is just like right in the mic.
Like he's busy at the moment.
Good job, Jake.
Yeah, nicely done.
Nice.
Yeah. Good job, Jake.
Sorry about my breathing.
That's minor.
Yeah, that, yeah, there you go. Beauty. That's good. Did way better than I did on my getting those cores off the starboard side. I mangled it royally my first 10 times.
All right.
So that was 103.
Yes.
Oh yeah, sorry, I got it. Give me something to do over here.
It was right in front of me. I'm just
That
too
was
used to doing it.
Push core 5.
Yeah, Mongo's the backhoe.
All right, so we are going 300.
Hate it when I move your camera.
You can do it.
Can it? Doesn't annoy you? I should say
Does a
moving
little.
your camera.
It goes a little easier, I think, with the buttons.
Moving your camera. I should just say what a command I'm doing. Panning right, panning left,
All right.
panning up, panning down.
So we're going
300.
300.
So you see it's kind of mostly along contour. Bridge nav.
I think I have a lot of mud on the porch. Yeah. From landing like 10 times.
225 meters, bearing 300.
That ain't nothing.